Okay, welcome everybody. You've come through to this evening's webinar. Tonight, I'm really pleased to say we're joined by Professor Cathy Thornton. Cathy is Deputy Head at the Medical School here at Swansea University. So we're really privileged to be joined by her this evening and have an exciting webinar. Cathy's going to be sharing with us her webinar on cells, immunity and COVID-19, which is really topical at the moment. So those of you that are joining us by watching via record, if you do have questions following the session, you can email us at study@swansea.ac.uk. So we'll have the webinar with Professor Thornton now, and we will pause for some questions at the end with those of you who are joining us live. So Professor Thornton, over to you.
Okay, thanks a lot, Emma. I will start by sharing my screen. Okay, so Emma's already done a very lovely introduction to me. So I thought I'd just start by sort of telling you that I teach our graduate entry medical students. So some of the content that I'm going to share with you today has been used very recently with our first year cohort, and that I also teach our undergraduate BSc programs in genetics, medical biochemistry, basically all of our undergraduate BSc programs, applied medical sciences, medical pharmacology, and our population health degree. And again, some of the content is going to be what I've taught our second years and our third years in the past few weeks and into the sort of latter half of 2020. So I thought I'd just try and introduce you to some of the basic concepts of our immune system and talk about some of the features of it that we may have been thinking about or been exposed to through the media, both the print media, the television media, social media, about the importance of our immune response in the current pandemic, but also give you a sort of flavor of the general importance of the immune response as well. And we've kind of been doing this with our students as well as trying to make sure that everything remains, relevant to human health and disease, because we have lots of people with a particular interest in pursuing that for their career post-university. So I think most of us will be familiar with what does the immune system do. We've all heard about antibodies and T cells more than ever before in the last year, and they are key cellular components of our immune system. And so our immune system has essentially evolved to defend us against us as multicellular organisms from pathogens. And we're going to talk about what some of those pathogens are. And I think we're all very familiar with this now. We're very familiar with the concept of needing to defend ourselves against viruses. A couple of years ago, we were particularly concerned about Ebola. Every year, we're worried about influenza. And at the moment, we're particularly worried about SARS-CoV-2. So there's a huge range of viruses of the respiratory tract, of the gastrointestinal tract, and that target other organs in the body that we need to defend ourselves against. And we can see the consequences of not being able to sufficiently defend ourselves against with, the pressures on the NHS at the moment. But of course, we mustn't forget that we're also constantly bombarded with different bacteria. We have our commensal bacteria, the ones that live naturally in our bodies, that don't really activate the immune system in a way that causes us any concern. But we do have other bacteria that we get exposed to, and we do have to mount an immune response to those. And you can see the example here. Here's an example of acne. So when you have those really raised, inflamed, potentially pussy acne on your face and on your back, for example, that's typically because of the immune response to the bacteria that are infecting the acne that is driving that particular feature. And that's why we can sometimes treat it with antibiotics and sometimes with some anti-inflammatories to combat both the bacteria and the immune response to that. And I always say to my students that if you see pus, it's just evidence of a really active immune response. And the pus is made up of all the cells in the immune response that have died after they've tried to do their thing as part of that immune response. And then we can see here the very unpleasant example of food poisoning. So many of us will have experienced food poisoning. Food poisoning is typically caused by the bacteria contaminating the food or the toxins that the bacteria have released and contaminating the food. And then we ingest them and all of the responses downstream of that. And that's our immune system that helps us combat that and clear that away. Then we have some examples of fungus here. And again, most of us will be familiar with people who've experienced this white tongue, which is a fungus called Candida. So this is candidiasis. And you can see a tissue section here in the middle, sort of the purpley things here are the nucleus of our cells, and the outer lines here are our cells. But these sort of dark pink structures here are the actual bits of the fungus that are inside our tissues and causing this problem and activating our immune response. And we can see the examples here of sort of ringworm and athlete's foot, slightly gross pictures, I agree. And then, of course, parasites, another pathogen family that is particularly problematic, particularly in certain parts of the world, and maybe something that for most of us who live in the UK only have to think about when we're going on holidays, if we can ever do that again. So here's the example of malaria. It's probably the one parasite that people are most familiar with. We have lots of different helminthic worms. And here's an example of a disease called lymphatic filariasis, where the worm infects the blood vessels and blocks the blood vessels and the lymphatics of the lower limb and prevents the flow of liquid back up through the limb and causes that accumulation of liquid that we see characterizing this filariasis disease. So yes, I think we're all very familiar with the concept that our immune system is really designed to protect us against this sort of pathogenic burden in the world around us. But our immune system does a whole host of other things. It has this classic association with defending us against pathogens, but it also has a really important role in regulating lots of other processes within the body. And if these become dysregulated, these will become associated with disease. So I'm going to introduce you to some diseases that are classically associated with the function or the dysfunction of our immune system. And you'll spot that they're not all diseases. Some of them are just normal functioning at different stages of our life. So autoimmune diseases, the kind of clue is in the name. It's where the immune system is now attacking ourselves. And many of you may have these diseases. You may have family members who have these diseases. And we've got some examples along the top here. We've got rheumatoid arthritis, and this is the classic appearance of the hands of someone who has rheumatoid arthritis and the damage that is done to the joints here and the disfiguration that occurs in the joints and the loss of capacity and utility of those joints. And this process is driven by our immune response. Our immune response, for some reason, has decided to attack our joints. And all of the cells and other mediators that are typically associated with the immune response against a pathogen are now targeting the joint. And you can see in this little cartoon here, all of these little cells here and these different mediators being made by these cells have a really important role. And I'm going to introduce you to a couple of those. And here's an example of myasthenia gravis, where we now make antibodies to one of the receptors at the end of our neurons. And so that the signal one neuron sends to another neuron doesn't work properly because the receptor that's going to receive that signal is blocked by the antibody. And we want antibodies that block things in some settings, for example, SARS-CoV-2 vaccinations and natural immunity, but we don't want it blocking a natural function in our bodies, or we will see dysfunction and association with disease. And another disease I think people may be familiar with that has an important contribution of the immune system is cancer. So you can have cancer of your immune system, and that's a little figure down here on the right. So leukemias, lymphomas, and myelomas are all cancers of our immune system. So different cells in the immune system are the cells that bear the cancer. But our immune system is really important in protecting us against cancer, and our immune system is constantly surveilling the cells of our body to see if any of those cells have developed the abnormalities that we would associate with cancer. And then the immune system would ideally remove those transformed, we call them cells, and prevent them proliferating and growing in number and becoming a tumor, for example. And so what we see in someone who progresses to having a diagnosis of cancer is that the cancer cells in their immune system are in a battle for supremacy, so to speak. And if the immune system is being sort of downtrodden by the cancer cells, then the immune system is unable to mount a response that will protect you against the cancer. And we can use lots of therapeutics these days, which are designed to boost the immune system. We call these immunotherapeutics, and they target this sort of war between the immune system and the cancer cells and rally the immune cells into fighting the cancer and hopefully leading to a remission of disease and hopefully cure. So there's some examples of diseases, but also our immune system is really important in the everyday setting of pregnancy. Pregnancy is not a disease, it's an everyday occurrence. And the woman's immune system undergoes a huge range of changes over the course of the pregnancy to help that pregnancy be successful. And some of the adverse pregnancy outcomes such as gestational diabetes and miscarriage are associated with dysfunction of the immune system, which then lead to the loss or adverse effects on the pregnancy. So again, you can see in this little cartoon, the same types of cells and the same types of mediators being produced by these cells that are really important in this normal phenomenon of pregnancy. And we also have a growing understanding of the contribution of the immune system to mental health disorders, particularly mood disorders, where we see these same cells and these same mediators produced by these cells and these mediators that circulate around in our bloodstream that can get to the brain and affect the mood in the brain and manifest as depression and anxiety. And there's a subset of mood disorders that we think can now be treated by targeting the immune system in particular. And so lots of interest now in progressing those therapies so that more and more people can be helped with their mental health disorders. So they're the kinds of things that our immune system is doing. It's helping defend us against pathogens, but it also has a role in a whole host of other diseases and normal physiological function that are important in our modern world. So what is our immune system? So we're talk about what it does. What is our immune system actually made up of? We've alluded to the role of different cell populations, but how does this all fit together inside our bodies? So we tend to now think of our immune system as having 3 different layers. And if you think about your own body for a moment, we can help to start understand what those 3 different layers are. The first layer is the layer that we can see. So if we look down at our skin is the first layer, one of the first layers of our immune system. And then all our other surfaces inside our mouth, inside our nose, inside our gastrointestinal tract, inside our genitourinary tract, all of those surfaces of our body are a really important part of the immune system because they act as anatomical, physical, and chemical barriers to pathogens, for example. Landing on those sites, we have defense processes to stop them getting a foothold and hopefully not even initiating a full-blown immune response. They're gone and away before our immune system has had a time to properly up and act in response to them. If we then have some kind of breach of those barriers, because those barriers are dysfunctional for some reason, there's lots of cells present in these barriers that are going to respond to that breach. That breach might be a pathogen, but that breach might be something else that our immune system detects as dangerous and is going to mount a response to it. So we have these cells that are really important in making that first response, and their role is to amplify and initiate the immune response and hopefully combat whatever has elicited that immune response before it has, again, a chance to get a foothold and cause us long-term problems. But we kind of have an underpinning safety net, which is not present in all animals. It tends to be only present in higher vertebrates, such as mammals. And we have what's called this adaptive immune system. And this is the part of our immune system which can learn about the environment around us. So this is the part of our immune system that when we have a second infection with a pathogen, this is the one that says, I've seen this before, and I'm kind of ready, armed and ready to go, and I'm going to defend the body against this particular pathogen. And that's what we're trying to do with a vaccination, for example, is we're trying to ramp up this adaptive feature of the immune response. We're trying to do it artificially with a vaccine so that when we have a natural exposure, the immune system is like, well, hey, I've seen this before, off I go, I've got antibodies, I've got T cells, I've got all of the things armed and active that I need to effectively defend me against this pathogen before the pathogen can get a foothold and cause fulminant disease. And so when we're thinking about these different layers, I'm going to give you a couple of examples of those physiological barriers, these anatomical barriers. I'm going to give you a couple of examples of the cells, and we have our soluble mediators, all of these things that the cells produce and release into the environment around them, and the things that are then found in our blood and other body secretions, like we find them in our saliva and in our nasal fluids, in our urine, in our tears. If you're a breastfeeding mother, you will find it in the breast milk. So lots of our body fluids have these soluble factors in them, which are critical components of the immune response. And you can see, for example, people might take saliva to try and measure antibodies for SARS-CoV-2 to sort of assess someone's immunity in the COVID-19 pandemic. So this is some examples of our anatomical and physical barriers. And again, just think of your own body. We have our tears, and they wash away anything that may be irritating in our eyes. And there are enzymes in those tears that are going to hopefully kill any bacteria that might try and establish a foothold in the eye. We have this on our skin. We have the sweat and the oil and the hair. We have a local commensal bacterial flora on our skin. All of these are designed to stop pathogens getting a foothold. We have these bacteria also living in our large intestines and in lots of our other mucosal surfaces like our respiratory tract. Our saliva is a really good example. It's hopefully washing away the microbes that might try to colonize our, around our teeth and in other parts of our mouth. I'm going to show you an example of the cilia in the respiratory tract. Our stomach, the acid in our stomach is great for digesting food, but it also kills any organisms that may get that far. And our bladder, just the fact of urination, is going to wash away microbes from within the urethra and again stop them getting a foothold and causing us an infection. So these are our anatomical and physical barriers. And again, like I said, they're really quite easy to visualize because we're all very familiar with our own surfaces. So here's an example looking at skin, and we can see the very outer layer of our skin drawn here as a little cartoon. And we can see these different layers of cells that are all interacting to give us this nice protective layer. And this layer is there to keep water in and keep the pH low, keep the skin healthy and that barrier intact. And this barrier of cells is accompanied by the sweat and the oil and the hairs of our skin to provide this really good barrier function. And the advantage of thinking about this from the perspective of the skin is we're all very familiar with what poor barrier function in the skin looks like. So here we have poor barrier function, and poor barrier function in the skin because by a whole host of things. We've all scratched our skin and seen the consequences of that. Different microbes landing on our skin can have an effect. Pollution can have an effect. Stress can have an effect. And we've got the example here of allergens that can have an effect, things like pollen or house dust mite or cat dander, some of the things that many of us will be allergic to. And there can also be some genetic variants in the human population that can contribute to poor barrier function. And the classic example of poor barrier function in the skin is eczema. And again, I think, many of us will have experienced eczema either, very acutely, or some of us will have suffered it chronically, perhaps when we were younger, and some of us may still suffer it into our youth and into our adulthood. But it's a really good example that's easy to visualize as impaired barrier function in the skin. And I've just got a couple of pictures there of some children looking really miserable because of their eczema. They've got impaired skin barrier function for any one of a number of reasons, and it has led to this really classic eczema response, which is a hallmark of that poor barrier function within the skin. But the other features of the immune response also contribute to this, and I'm going to bring us back to a couple of examples of that as well. And so another example of barrier function, particularly important in terms of SARS-CoV-2 and COVID-19, is the mucociliary clearance processes within the respiratory tract. And so the surface of our respiratory tract is covered in a sort of fine mucus, and the cells that line the surface of the respiratory tract have these little beating cilia on them. You can see that in the diagram here, these little hair-like structures that stick up, and these beat and they try and shift anything that's caught in the mucus back up through the airways. Then when we cough or sneeze, out they come. And so this coordinating beating of these cilia expels any pathogens that are trapped, or any toxins that are trapped, and expel them from the respiratory tract. And the cells here are also producing a whole pile of other factors, such as these antimicrobial peptides that are also helping us defend ourselves against any pathogens that have penetrated into the airways. And on the next slide, I've just got a little example of those cilia beating so that you can have a break from my voice and hear this very nice, I think, American gentleman.
Cilia, like the cilia depicted here, are found on the cells which line the nasal cavity, the larynx, and the trachea. There is mucus secreted by goblet cells in these respiratory passageways. And this mucus collects foreign particles, whether they be microbes, from sneezes and from the air, also diesel particles, dust, soot, etc. All of these foreign particles are caught in the mucus. And then the cilia help to sweep the mucus away from the lungs. They sweep in unison towards the stomach, because this mucus then reaches the throat and we swallow it involuntarily into the stomach. And since the stomach is a big bag of acid, this is a far better destination for germs and toxins and dust than would be the very delicate lungs. And so these cilia sweep foreign particles away from the lungs and towards the stomach, where it can be disposed of.
Oops. Okay, so that's thinking about the surface of our body. And if we sort of start digging below the surface and thinking about those cellular and tissue components of our immune system, because I think we're all very familiar with the heart and its role in the cardiovascular system, and the brain and its role in the central nervous system, but I suspect we're all a little bit less familiar with the sort of organs and tissues and cells that are part of the immune system. So this is a little cartoon of the human body and highlighting the different parts that we consider to contribute to the immune system. And the one that we're most familiar with, I suspect, are the lymph nodes. So the lymph nodes, we have them dispersed all around our body, but the classic example are your tonsils. And we all know, you know, our mums have done it to all of us at some point in our lives. We said we're a bit poorly, we've got a bit of a sore throat, and they've gone in and felt in under here in your neck, looking for these swollen glands. And so these glands are the lymph nodes, and these lymph nodes are just little bags of immune cells. And the reason they swell when we've got an infection is because our immune system is really active. And so there's all kinds of fluids flooding in there. The cells in there are proliferating, they're increasing in number. One cell is dividing into two cells, and those two cells are then dividing onwards and onwards. And so we're increasing the number of cells, and there's lots of fluids passing through there. So of course they get bigger, and we can really feel those. But we also have this little organ that sits on top of our heart called the thymus, which is really important in the development of that population of cells called T cells. And then we have the spleen here, which is an important contributor to the immune system as well. All of the cells of our immune system are made within our bone marrow. So we have these cells in our bone marrow called hematopoietic stem cells, and these give rise to all of the cells of our immune system. So if you think about the concept of a bone marrow transplant, what we're doing when we're doing a bone marrow transplant is taking those hematopoietic stem cells and giving them to another donor so that they can reconstitute a healthy immune response with those hematopoietic stem cells from a healthy donor, having destroyed the immune system of the unhealthy donor through radiation therapy, for example. And then the other thing is we have obviously more of these are just examples of lymph nodes, but these are the lymph nodes we can't feel. These lymph nodes line our gastrointestinal tract. And these are, you know, called Peyer's patches, and there's other types of these, and these are lining our gastrointestinal tract and are really important in providing immune protection all the way through that particular structure. So these are all the sort of different tissues and components that make up our immune system. And those hematopoietic stem cells that we find in our bone marrow are going to produce a whole host of different cell types. And these cell types are going to be found in different tissues. And when there's a signal in that tissue that there is some kind of danger, some kind of damage, then those cells are going to move into that tissue and they're going to try and remove that threat. They're going to activate the immune system in that process. And then hopefully it's all going to be tidied away at the end and you restore healthy function to the tissue. And I've used this example here because we've talked about skin barrier dysfunction and its association with eczema. And I was using this slide yesterday to teach our third year AMS. We were looking at ethnicity and the differences in this case between the response in the skin of a black person and the skin of a white person. And you can see the little pictures on the side here of what the disease looks like on the skin of a white person, on the skin of a black person. And when you can take samples of that skin. Again, skin is really nice to work on from the perspective of getting access to it to do your experiments and your research. And you can take skin from a healthy person. Here's a healthy African American, and here's a healthy European American. And then you can take skin from someone who's got eczema, and you can take the skin from healthy skin on that person where there's no eczema lesions, or you can take the skin from right inside the eczema lesion. And then you can do some experiments on it, and you can look at the cells that are present in the skin, and you can start to understand there's a disease process that's going on. And in understanding the disease process, you can hopefully develop some nice therapeutics to interfere with that disease process. And so you can see here, we don't need to worry about the different types of cells along here, but the key difference is in the healthy skin here, on the African American, on the healthy skin on the European American, we don't see a lot of immune activity. There are some immune cells in there, and these are the cells that are really important in surveilling our environment all the time. And they're the cells that are going to initiate the immune response if there's some kind of threat. But you can see there's a perception of threat, and there's a whole host of cells have invaded into the skin and are driving the immune response in the skin. And that immune response is then manifest as these symptoms of eczema on the skin. So if we look at these little slides here, these little pictures, here's the skin, here's that sort of epidermal layer, that very outer layer of the skin, and this is the layer underneath, and all these little brown dots on each of these slides are different types of cells that have infiltrated into the skin. And we can enumerate those to understand what's going on and which different cell populations are contributing to disease. And we can study each cell in more detail to see what kinds of things it's making that are contributing to the immune response. So these mediators that it's making will be particularly good targets for treating the disease. So we can see these cells are moving around our body and moving into tissues in response to some kind of insult in that tissue, and then contributing to the immune response, which is going to hopefully clear away that insult and leave you with normal, healthy skin at the end of it. But clearly, this sometimes goes wrong. We fail to downregulate the response at the end, and then we get inflammatory diseases like eczema and diseases of other organs as well. And so I've mentioned a lot these mediators our cells are producing, and these mediators our cells are producing are the way our immune system talks to itself and to other cells in their body. So we have our cell of the immune system here shown as this blue cell, and it talks with other cells in two basic ways. One, it releases all of these soluble mediators shown here as these little dots, and these mediators come out of that cell, and surrounding cells or cells far distant from this, in another organ in the body, because these can pass through the circulation, will have on it a receptor for that, which enables it to recognize that little soluble mediator and initiate a response to it. So these little mediators, these little dots, are kind of the words that the immune system uses to communicate with the cells around it and elsewhere in the human body. Instead of releasing these mediators, the cells also express lots of different molecules on their surface, and these molecules can interact with molecules on the surface of other very nearby cells in this case, and that can help the cells talk to each other and initiate and amplify the immune response that we need to drive the defense against whatever the insult was that caused the immune system to be activated. So SARS-CoV-2 being a really good example of that. And this is a way in, looking at the communication mechanisms of a particular type of cell. I chose the eosinophil because it's one of my favorite types of cells. And the eosinophil, we find them in our blood, we find them in our tissues, and they're full of these structures we call granules. And these granules are full of these mediators that are all preformed and sitting in the cell, waiting for the cell to get a little tickle from some kind of environmental insult. And then they're going to disgorge those granules. They're going to vomit out these granules into the surrounding environment and release all those mediators. So here's a little cartoon at the top of what those eosinophils may be doing with those granules. They can release them in a whole host of different ways, which we don't need to worry about. But the fact is they release them from out to the outside of the cell. They don't take the bacteria or the pathogen into the cell. They release the contents of these granules into the surrounding tissue environment. And I've shown down here this picture of these cells under an electron microscope. So the arrows are pointing at their mitochondria, but you can see these other structures here. These structures here are these granules within these cells. And I've shown this diagram because this is from some research in my own group. And the first author of this research was someone who had done their undergraduate degree at Swansea, had gone on to do a PhD, and is now one of our lecturing staff and a really active researcher in the medical school. So sort of he's gone on the journey that many of you are about to start on, and has sort of got to the point that he aspired to, and is now going on to make a teaching and research contribution to the university. But you can see this this microscopy work, this electron microscopy work is done, and you can see these nice granules in here sitting inside these cells, ready to disgorge their contents should they get some kind of signal that suggests they need to release their contents. And the classic signal that's going to make an eosinophil release its contents is some kind of parasitic worm. And then you can start to see the difference in the size of the eosinophil versus the worm and start to go, that's why they have to release their contents. They've got to dump their contents onto that worm and try and destroy that worm. And so I've got another little video of you showing these eosinophils put in a test tube with the helminthic worm, and then the response they're going to make to try and destroy that worm. And so you can see the eosinophils are starting to shake. They're getting signals that they've got to move towards the worm. They all start moving towards the worm. And then once they get to the worm, they're going to degranulate. They're going to release all those granule contents onto the worm. And those granule contents are full of enzymes that are going to hopefully attack the worm, and full of other mediators that are going to bring in other cells of the immune response and hopefully destroy that worm before it gets a chance to give you a parasite infection. And I've mentioned a couple of times that we have these insults and eliciting agents that attack our immune system and get our immune system going. And we think of these as in two broadly relatively similar categories, but they have very different origins. So this concept of anything dangerous is going to initiate an immune response. And that danger can come from a pathogen. And that's our classic understanding of how the immune system is activated. The pathogen has these structures on it that our immune system has evolved to recognize. And then our immune system sees those and goes, okay, I need to mount a response, and I'm going to initiate and amplify the immune processes and hopefully destroy this pathogen again before it can get a foothold and cause us an infection. But we can get these danger messages from other sources as well. And a classic is when our own cells are stressed or damaged in any way. So if our own cells become infected with a virus, they're not only responding to the virus, they're responding to the havoc that that virus is wreaking within our cells. And they're releasing other mediators that are activating the immune system and saying, oh, my cells are not particularly healthy. I need to try and repair those unhealthy cells and restore normal function to that tissue where the dysfunction is occurring. And again, a really good example of that is coronavirus and lots of other respiratory viruses. So here we've got the epithelial, a little cartoon of the epithelial cells that are lining our airways. And here we've got our good friend SARS-CoV-2 that's infected those cells. And it's infected those cells, and the cells are going, virus, I must mount a response. And the cells are being damaged. And so they're releasing other mediators that are indicating to the immune system that there's a viral infection and that the cells are damaged. And that signal will be coming from any cell that is infected with the virus. And so these cells are now releasing these soluble mediators out into the local environment. They'll be coming out into the airways, but they'll also be making their way through the vasculature, through the bloodstream, from the lungs and down through to other organs in our body. And that will attract in the immune cells that we need to mount a response to destroy those virally infected cells and stop the viral infection spreading from one of our cells to the next of our cells, and so on. And one of the cell types that will be activated by that is a cell type called the natural killer cell. And what the natural killer cell does is the natural killer cell is really primed to identify our own cells that are a little bit unhealthy. So they're really important in that early response to a cancer cell. So a cell that has changed from a healthy cell to a cancer cell has different molecules expressed on the surface of it. And the natural killer cell can differentiate between the healthy cell and the cancer cell and try and destroy the cancer cell before it can proliferate. And it can do the same with a virally infected cell. It can see these receptors on the surface of a virally infected cell are different to a not virally infected cell, and it will try and destroy the virally infected cell before it can spread the infection to the neighboring cells. And it will do that by the release of these little granules of materials here onto the infected cell. Again, these enzymes and other really active mediators, which are hopefully going to kill the virally infected cell or kill the cancer cell and stop the infection or stop the progression of the cancer. And so we can see why studying these cell types are really important in a number of different settings. And thinking about coronavirus in particular, I thought I'd just go into a little bit of depth about the response again, because there's so much interest in this and so much information out there. You know, we watch the news every night, and every night there'll be something about the immune response. You know, it may be a horrible thing to say, but it's a very good time to be an immunologist because there's a greater understanding in the community about the importance and function of our immune system. So here we have the example of our virus infection. And so viruses are just little packages of protein, DNA, and RNA. And the cells in our immune, or the cells of our body, all the cells of our body, have receptors in them that are able to identify this foreign DNA, this foreign RNA. It's not my DNA. This is DNA from somewhere else, or RNA from somewhere else. And I'm going to mount a response to that because it's foreign, it's dangerous, it's an insult. And so there's a whole host of intracellular signaling pathways which are going to take the signal from those first recognition of the virus through to our DNA, and it's going to switch on lots of different genes, which are then going to amplify as proteins and amplify the immune response. And there's a family of particular genes that are switched on, and these are called interferons. And again, this may be something that some of you will be familiar with, depending on, you know, how much you're delving into the literature at the moment around this. And so this interferon is really important in driving that immune cascade, this amplification of the immune response, and bringing in all those mediators and cells into the respiratory tract and trying to combat the virus before the infection becomes too serious. And so the cells are responding to this interferon, and this interferon does a number of things. One thing it does is it interferes with the virus's ability to replicate itself inside the cell. So it's trying to stop the virus replicating, and that will stop the virus spreading to neighboring host cells, and it's going to drive the amplification of that immune response. It's going to switch on genes and switch on proteins that are going to bring those NK cells that I introduced you to on the previous slide into the environment where the virus is, and those NK cells are hopefully going to destroy any of our cells that are infected with the virus. And so these two really important features of this early cascade effect in the immune response will hopefully limit the disease course for most of us. But obviously, this was one of the things people started looking at very early on when we discovered that people would be getting very severe COVID-19. And one of the things that was found is a paper from very early on, and they found that people with severe COVID-19 had a particular impairment in this interferon pathway. So they were either not very good at producing the interferon, or they were not very good at responding and amplifying an immune response driven by the interferon. And so you can see if their interferon pathway is not working very well, they're not going to be able to inhibit viral replication, and they're not going to be able to initiate and amplify those early immune responses. And so therefore it becomes not surprising that they progress to having a more severe form of disease. And so just sharing the last few slides with you and touching on the sort of other immune responses we're making to something like SARS-CoV-2 coronavirus, we're all very familiar with the concept of antibody and huge interest in, do we make antibody? How much antibody do we make? Is that antibody long lasting? Are there antibodies able to respond to different variants of the virus? And we can see that the reason we're interested in this is because one of the things the antibodies do, these little sort of pink, so it's an inverted Y-shaped structure here. One of the things this antibody is going to do is it's going to bind to this spike protein on the surface of the virus and stop the virus then being able to infect our cells. It interferes with the ability of the virus to bind to our own host cells. And therefore, if it can't bind to our host cells, then it can't infect our host cells and develop and establish an infection. And that's the process by which it will then protect us from having disease. And so we can see lots of studies here looking at these antibody responses that people make in response to natural infection with SARS-CoV-2, and of course immense interest in the response that we will make with antigens. And you can see this sort of these patterns of antibody changes over the course of infection tell us a little bit about the infectious process and a little bit about the individual response to that. And we can also use it in screening programs as we are doing to identify whether someone has established immunity to it, to SARS-CoV-2, by measuring this antibody that our immune system has made as part of that response to that virus, that response that initiates in the respiratory tract and then is disseminated through the body to key organs of the immune system. And so one of those key organs are those lymph nodes that we have dispersed all around our bodies, because these lymph nodes are really active sites of immune activity, as I've already explained to you in terms of the tonsils. And so what's happening in there is that we have these T cells and these B cells that are critical components of our long-lasting immunological memory response to any kind of infection. And so these lymph nodes have these really highly organized structures where the cells can be best brought close to each other and initiate these antibody and other responses, these T cell responses that are also really important in our lasting immunity to SARS-CoV-2 and other organs, other diseases. And so you can see these structures here, within here, and we have different types of cells that are preferentially located in different regions. And they're really important in terms of the sort of life cycle of establishing that adaptive immune response, that specific immune response, that immunological memory to a pathogen, be it a natural exposure or be it a vaccinated exposure. And the last two slides I want to share with you, just highlighting some of the issues that we've been discussing around SARS-CoV-2, I would say around water coolers, but most of us aren't at work, but maybe in our homes, maybe with friends via Zoom, is this interesting effect of aging and the susceptibility of older people to the detrimental effects of SARS-CoV-2 and other infections. We know influenza, serious influenza is more prevalent in the elderly. And we have this concept of immune senescence. So our immune system is changing as we get older. When we're younger, much like many other features of our bodies, everything's much more robust. We have a much more robust immune function. We make a better response to infections. We make a better response to vaccines. And what happens as we get older, our immune function declines. So now we don't make such a great response to vaccines. We don't make a very good response when we get infected with something new. We don't even make a very good response when we get infected with something that we have immunological memory to, because our immune system is kind of waning through the sort of post 60 years of age part of our life. And so this becomes very important in understanding differences in disease processes and susceptibility disease. And it's this change in our immune system, which is also why we see increased onset of malignancies related to cancer, for example, and increase in a whole host of inflammatory diseases like autoimmune disease, cardiovascular disease. It's a changing immune system and the accumulated effects of the environment we've been exposed to during our early decades. So that's the effect of aging. And we try to take this into account during all of the studies we do in my research group, for example. And the last thing I want to end on is obesity, because again, lots of interest in the effects of obesity on the vaccine response to SARS-CoV-2 and the susceptibility to getting severe COVID-19 or dying from COVID-19. And what happens in obesity is a dramatic change in our adipose tissue, which we can all visualize as someone who's increasing body mass index. But not only is there a change in the sort of fat cells within the adipose tissue, within the fat tissue, the immune cells within that adipose tissue are also changing. They're changing and they're becoming much more inflammatory, much more activated. And the consequence of this is there's spillover into the circulation and out of the adipose tissue into our other organs. And this affects the function of our immune system and our endocrine system and our nervous system. This spillover of this inflammatory signature from the immune cells and from the fat cells within that increasing volume of adipose tissue. So that's why we see changes in the immune response to things like SARS-CoV-2, in addition to some of the sort of physical effects on respiratory function, for example, that we may see in someone who was morbidly obese. But it's these fundamental effects of obesity and age on our immune system, which we're really only starting to come to understand. We can see really in action during the COVID-19 pandemic with these particular populations of people susceptible to severe disease and COVID-19 and mortality from COVID-19 because of these underpinning changes in their immune system. So hopefully that has given you a little bit of a taster introduction to the immune system, focusing, where I could on sort of topical things related to COVID-19, but I'm happy to take any questions as well.
Fantastic. Thank you so much, Professor Thornton. That was really, really informative, and I know so valid and relevant to those that are joining us this evening. So thank you so much. Before we open up the floor to questions, as we do want to hear from those of you that are joining us, just to say, those that are watching this on record, thank you for joining this evening. As we said at the start, this is part of a series of our webinars taking place over the next couple of months. So you can register for those on our webinar page, and we will be sending out a link to everyone, all our offer holders and inquirers, to book on to future sessions. So I'm just going to cancel the record and then we'll start with the Q&A. So thank you for joining us and watching on record.